Hi, my name is Anjali Agarwalla. I am a rising junior at Barnard College. I'm a neuroscience and behavior major with a cellular concentration. I was working in Professor Glendining's lab in the biology department at Barnard College. And my research this summer was on developing a rat model for pharmaceutically induced taste distortion in chemotherapy patients. So chemotherapy, while extremely beneficial for cancer, has been known to have many negative side effects, and amongst these is actually 68% of chemotherapy patients reporting altered sensory perception in the way that they perceive taste stimuli and smell stimuli. And this is a huge issue because as a result of this, 40% of cancer patients suffer from malnutrition, and 20% of cancer patients are actually citing malnutrition as their primary source of morbidity. It's a huge phenomenon that's occurring in the oncological world, and unfortunately we don't actually know what's causing it. There are a few proposed mechanisms, including damages in sensory receptor cells, oral mucositis, but we don't actually know if it's one of these mechanisms, all of these mechanisms. It could be none of them at all. So the goal of our research was to develop a rat model where we simulate the same taste distortive effects in rats so that we were able to isolate exactly what's happening when patients are given chemotherapy and why it's leading to such changes in their peripheral taste system. So we were working with a pharmaceutical company called Novartis, who have recently released a drug called Compound X, which is very effective in chemotherapy, but has been showing a lot of reports of taste distortion. So we took rats and we dosed them for 2 weeks with this chemotherapy drug. And at the end of the 2-week dosing period, we then analyzed their changes in behavioral responses to aversive stimuli and appetitive stimuli, basically stimuli they would like to eat and stimuli they would not like to eat. So when we were analyzing this behavior, we had a few questions we asked. One of the first questions was, how does drug treatment affect the way the rats experience taste stimuli when they are exposed to multiple taste stimuli in the same time period? For example, in the same 30-minute trial, giving the rats both MSG and sucrose at different concentrations. And then similarly giving them citric acid, NaCl, and quinine, which are aversive stimuli, in the same time period. So we found that when we give them appetitive stimuli, which are things they would like, such as sucrose and MSG, there was actually not a discernible difference between the way the control rats and the experimental rats licked at these solutions. However, when we gave them aversive stimuli, we found that at high concentrations of salt, there was a decreased aversion, and at high concentrations of quinine, there was an increased aversion. That is to say that rats that were given the drug were licking more avidly at NaCl than rats that were not, and were not licking as avidly at quinine as rats that were not given the chemotherapy. So our next question is, what happens when we give the rats a single taste stimulus? So for example, instead of giving them both NaCl and quinine, giving them only NaCl or only quinine at varying concentrations during that time period. And we found that there was not the same response. We weren't seeing any difference between experimental and control animals when presented only with a single stimulus, despite the fact that when presented with multiple, we were seeing a significant one. So what does this mean? So our current question is, what's causing this specificity in behavior? Why are we seeing rats only respond in the presence of multiple stimuli? So one hypothesis is that human chemotherapy patients are not only eating NaCl and they're not only eating quinine, they're being given full meals. And in being given full meals, they're being presented with multiple stimuli at the same time. And this presentation with multiple stimuli implies that there might be a context dependency for this taste distortion, that it only really occurs when there's multiple stimuli being presented to the patient. So our one question would be how to simulate this context dependency in the rat model to be able to isolate peripheral changes. Furthermore, we're actually only testing one of the 3 systems that's implicated in classical taste, which is how the flavor of the food is on your tongue. There are 2 other systems, the olfactory, how it smells, and the somatosensory, whether or not it feels good in your mouth, if it's irritating, if it's painful, if it causes any change in temperature. So further experiments would be done into testing stimuli that aren't just activating your taste receptors, but also activating olfactory receptors, such as ethanol, or activating your somatosensory receptors, such as capsaicin, which causes a burning sensation in your mouth. So that would be one of our future experiments. Furthermore, we want to test the peripheral nerve recordings of these rats, expose the nerve, and test how the rats are responding to stimuli being passed over their tongue at the nerve level as opposed to just at this receptor level, to see if there's an issue downstream of the receptors. So hopefully, in doing these further experiments, we'll be able to isolate exactly what's happening in these rats and analogously what's happening in their human counterparts, and be able to better create medicines that won't cause this incredibly deleterious side effects of chemotherapy.